Hello biology students, and welcome to video 7, the last video in the cell unit. So in this video we're going to do two things. First, we're going to finish our discussion of passive transport, which remember is the transport of molecules across a membrane not using energy from high to low concentration. And then in the second half of the video we'll jump into active transport. So first, let's finish up passive transport. So as a reminder, when we looked at there, we've seen so far two types of passive transport. We've seen diffusion, which was when small nonpolar molecules diffuse freely across the membrane. And we also saw osmosis, which was an example of when a polar molecule, water, crossed the membrane from high to low concentration of water using special channels called aquaporins. So because water is charged, it had to go through a channel to do that. However, there are other large or polar or charged molecules that need to diffuse, that have to get across the membrane but can't just pass freely. So that's the situation we're going to look at last. The last kind of passive transport is facilitated diffusion, when we get large polar or charged things to diffuse across the membrane using a protein pore. So facilitated diffusion is the movement of specific molecules that are polar or large, so things like maybe glucose or amino acids, down a concentration gradient, so from high to low concentration, using a transport protein. So there are two ways this can happen. I can have channel proteins. So I have a picture of a channel protein right here. I just have an opening. It's never open or closed, molecules can just move down their concentration gradient through this opening. I can also have carrier proteins, which are a little bit different. So here's a picture of a carrier protein. So you can see here I have amino acids and sugars, so things that are too big and too polar to pass through the membrane. And I have a high concentration and a low concentration. And you can see they bind to the carrier protein. Them binding causes the protein to change shape and allow it to pass through, and then it closes again and the next one could bind. So this does not require energy because it's the actual amino acid or sugar that's binding that causes the conformational change in the shape of the carrier protein. So that's passive transport. Diffusion, osmosis, and facilitated diffusion all did not need energy. Diffusion was when things were nonpolar and small and right across the membrane. Osmosis was the diffusion of water, and facilitated diffusion was when we had a larger polar molecule that had to diffuse through a channel or a carrier protein. So then finally, to close this video out, we're going to discuss active transport now. And we have three examples: cell membrane pumps, like ion pumps, and endo and exocytosis. So active transport now is the opposite of passive transport. It requires energy, which, by the way, the energy molecule is ATP. And this makes sense. Active requires energy. If you're active, if you're working, you need energy. And we're moving molecules against their concentration. So now we're going from a low concentration to a high concentration. And you think this does make sense. Things naturally want to move from high to low concentration. Because if I draw a little picture here, so say I have a high concentration over here and a low concentration over here, and here's my membrane, I'm going to have more collisions on this side, so things are naturally going to move this way. So if for some reason I want to move from low to high, I'm going to have to work and use energy to do that, because I'm going against the flow. So you might think, well, why would you want to go from low to high? So maybe pretend that this is the cell, and these are nutrients. Even if I have lots of nutrients in the cell and only a few out, I still want to go from low to high because I want to get as many nutrients in the cell as possible. We can think of another situation where maybe I only have a couple of molecules inside the cell and lots outside, but say this molecule was waste. In this case, I'd also want to go, I want to go from low to high because I want to get waste outside the cell. But because things naturally want to move high to low because of diffusion, I have to use energy and actively use energy to get them out of the cell. So let's look at our first example of active transport, which is ion pumps. So here I have a situation where I have some ion. It can be any ion. Say it's a sodium ion. I have a low concentration on one side and a high concentration on the other side of the membrane. And say I want to get more ion going that way for whatever reason. So this looks a lot like carrier proteins, but it has one big difference. So the ions will bind to my protein, but that binding does not cause the protein to change shape. Instead, ATP, our energy molecule, will drop off a phosphate group, because ATP, that P stands for phosphate. So it drops off that phosphate group. And when that phosphate group binds, then the protein changes shape and lets the molecule go across. Now, dropping off that phosphate group, that takes energy. So we're breaking off part of our energy molecule to allow this to happen. So we're using ATP, we're using that energy molecule to pump ions across their concentration gradient. So here's another example. So you can see, here I have another ion pump. My ion binds, I use some ATP to allow a phosphate group to bind, which changes the shape of the molecule and it moves across. I mean, in theory, this could work both ways. I could then have a second different group bind, and before the phosphate group breaks off, that could allow the solute to pass through as well. So that's another example for you. Okay. So and then finally, our last example of active transport is what we use to move very large molecules, or big groups of molecules across the membrane all at the same time. And we're going to use vesicles for this. And the two examples of this are endocytosis. Endo means in. So endocytosis is when we're going to move lots of stuff into the cell. And exo, that kind of sounds like exit. This is when lots of molecules are going to exit the cell at once using energy. So endocytosis is when molecules are entering in bulk. So you can see that maybe I have all these molecules that I want to get into the cell. And maybe there's already tons of them in here. Now, imagine there are millions and millions in here. So I have a low concentration out here and a high concentration in here. But I want to get more into the cell because, maybe there are nutrients or something useful. So what's going to happen, the membrane is going to pinch in, form a vesicle, trap some of them, and bring them into the cell. Now, this process takes energy. It takes energy to form this vesicle. So this is an example of active transport. And because things are entering the cell, it's endocytosis. There are three types of endocytosis. If the things entering the cell are solid, so if the cell was eating solid things, that's called phagocytosis. So maybe if I had a cell that engulfed an entire bacteria cell, so maybe I have an amoeba and it engulfs an entire bacteria so it could eat it, that would be phagocytosis. White blood cells, WBC stands for white blood cells, they're the cells in our body that attack foreign invaders. So if these, engulfed a bacteria, that would also be phagocytosis. Penocytosis is kind of like cellular drinking. So if I transport a solution, so maybe DNA dissolved in water, protein dissolved in water, glucose dissolved in water, if I'm bringing that into the cell, that's penocytosis. And the third one is receptor-mediated endocytosis. And this is where I bring in something to the cell, but it's only one specific kind of thing. So I have receptors on the outside of the cell membrane that only bring certain molecules in the vesicle that forms. So let's look at some pictures so we can help ourselves understand these. So here I have phagocytosis. You can see this amoeba is forming a vesicle around this solid food particle. Here I have penocytosis. You can see the cell membrane is pinching to bring in this solution. Maybe it's a glucose solution or something like that. And then over here I have receptor-mediated endocytosis. So here's the membrane of the cell that's pinching in. And on this membrane are little receptors that are only going to let very specific molecules bind. So maybe only glucose molecules would bind. So when the vesicle pinched off, I would only be trapping in glucose. So this one is the most specific type. Okay. And finally, the last type of active transport is the opposite of endocytosis, which is exocytosis. This is when I'm dumping molecules in bulk out of the cell. Okay, so I'm going to get a vesicle that fuses with the membrane and releases the contents to the outside of the cell. So let's look at a picture. So here I can see an example of exocytosis. I have a vesicle. Maybe this vesicle is carrying waste. So what's going to happen? It's going to fuse with the membrane and dump its contents to the outside in bulk. And again, this takes energy to do all this work, so this is active transport. Here I have a pretty cool picture. This is an actual microscope picture showing the vesicle, bonding with the membrane and dumping all its waste to the outside. So you can see here, it looks very similar to the cartoony picture we have on the left. So that's it for the cell unit, and I'll see you all in the next unit.